First, we start out with an organism. Now that we have our tissue from our organism, we need to precipitate out the DNA. We do that with phenol and chloroform. This will precipitate out our DNA so we can start doing downstream analyses. Now that we have pure extracted DNA, we have to test to see if it's clean, high quality, and good amounts in order for us to do downstream analyses. In order to see the concentration of DNA within our extractions, we use a machine called the Cubit. This will tell us the amount of DNA that we have in our extraction based on a electrophorescence method. We also tested the quality of the DNA using gel electrophoresis. Now that we have high quality and high yield DNA, it is time for us to fragment the DNA into smaller pieces for Illumina sequencing and begin library prep. Now that we have finished fragmenting our DNA, we have to clean it up. Cleanups are going to take place throughout the library prep process, and the reason for this is that many of the reagents need to be cleaned up so we just have pure DNA. We'll use the Serapure beads, which the DNA will bind to, but none of the reagents will. We will be able to separate the DNA by using these magnets that the DNA is attached to the beads, and the beads will be attracted to the magnets. Once that is concluded, we will have pure DNA ready for our next step. So now that we have tested that our adapters have ligated and our fragments have fragmented to the appropriate size, we now need to actually select the size that we need in order for Illumina sequencing. The Blue Pippin uses a gel-based protocol in order to select the appropriate size in which we have selected for. In this case, we're looking between 438 and 538 length pairs. This will run for a half an hour. When done, we will have the appropriate length size of DNA in which we can start sequencing. Now that we have size selected, we have to understand and test to see if the appropriate sizes were found. So we use the bioanalyzer here, in which we load a specialized chip, and once that chip goes in, we will let it run and see if our size selection was actually successful. After our bioanalyzer run, we have found that the appropriate size of DNA base pairs is right in range between 438 and 538. On either side we have these tall peaks, those are our reference ladders, and we do have one issue with a smaller peak right around 120. Those are adapters that did not ligate, and so we need to clean the sample up one more time in order to get rid of those and only have our pure DNA at that second peak in between the large peaks that we see.
Next, we biotinylate our probes. Our probes are then hybridized to the DNA fragments. These fragments will then be used in order for us to actually do a hybridization and gather the DNA fragments of our specific choice. So that's why we have the probes. And as you can see here, what is going to happen is that these probes will latch on to the selected DNA fragments, and then they will be attached to streptavidin beads. These streptavidin beads will then be attracted through magnets again with the specific DNA fragments that we're looking for and targeting. Once these have been pulled out, we're ready for sequencing.
Now after all those steps, we have our final pooled samples prepped and ready for Illumina sequencing. Doesn't look like much, but this is the end work of our ion gauge, and it has lots of DNA ready for analyses. Now that our samples are ready for sequencing, here's the MiSeq where we will sequence. This will take a couple of weeks just because it costs a high amount of money and we don't have enough samples to fill a whole lane. If we did, we'd be wasting maybe a couple grand. So at this point, we're waiting for any other lab members who will then start sequencing, and we'll do it all together, saving time and money.